Yeah, we already talked about this guy. Um, he, yeah, came up with. It doesn't seem like it's that like exciting, but he was at the University of Kansas and developed this sort of equipment for creating very, very, very fine micropipettes, and then had a system, an experimental system that allowed him to use that micropipette in preparations on his microscope stage. So this was, yeah, 120 years ago. This happened. Um, these these Barber pipettes, as they're called, Barber micropipettes, were super useful. You could use them to inject stuff. You could use them for all sorts of very fine work on these microscope stages. However, this individual, a woman named Ida Hyde, 15 years later, took a Barber pipette and filled it with mercury and used this micropipette filled with mercury to stimulate the living cells of in Vorticella nebulifera, this little critter down here, and was able, using this micropipette, was the first person to actually demonstrate graded potentials using one of these micropipettes. This was a huge leap forward, and I don't want to call her like the most hidden figure. She's not, but she, reasonably well recognized compared to others, but still perhaps not as recognized as she should be. She was the first woman to get her PhD, her doctorate in natural sciences from Heidelberg University in Germany. And 13 years later, went to Kansas where Barber was. This is how she came in contact with Barber's innovation 5 years later. But she was a physiologist as opposed to Barber, and created the Department of Physiology in Kansas. Go Kansas. In 1902, she was the first female elected to the American Society of Physiologists. So Ida Hyde was a pioneer, was a real pioneer. I have this little quote from Ida here from a book that she wrote before women were human beings. So you can already tell from the title that she wasn't just a scientific pioneer, that she was also quite an activist and a feminist. Here's the quote. "It was not until I had worked many days in the splendid laboratory assigned to my private use that it dawned upon me that I was occupying a unique position. In the university circle, the news quickly spread that an American women's rights freak had the boldness and the audacity to force entrance into the college halls." So, yeah, it's a very interesting character. Maybe I have to put it on my reading list. I actually have not read Before Women Were Human Beings. I've just read quotes from it. So maybe we can read that together someday. But it's on my list of things I would like to read. Ida Hyde. Building on the groundbreaking work in a number of dimensions of Ida Hyde was this woman, Judith Graham, another American that was as a PhD student in the 40s, developed a potassium chloride-filled glass electrode. And this was a real step forward, right? Because, first of all, mercury's gnarly stuff. You don't want to really be messing with mercury a whole lot, and it's difficult to work with for a number of reasons. It's fine for conducting electricity, but having an electrolytic solution of potassium chloride was a major step forward. And that's why she's in this narrative here, because this potassium chloride micro electrode in a Barber pipette is set the stage for a lot of work that came later. But she's actually best known for a laboratory technique called cryoprecipitation. And she's known for this because this process of cryoprecipitation is how you're able to extract and concentrate blood clotting factors. This saved truly countless lives. Millions of people have been saved. Their lives have been saved by this technique that she developed. She was the first person to get a stable recording that was reasonably close to the contemporary value. What's the resting potential of a cell, of a neuron? 70, minus 70. She recorded that at minus 62. That was the first value that was actually within a reasonable range of being correct with these potassium chloride filled electrodes. Unfortunately, she died at a young age of 55, pardon me, from a brain tumor. So she had a sad end, and because of that, she was. I do not believe she was awarded the Nobel Prize. She never received. She certainly would have. The problem with the Nobel paradigm is that it's not awarded posthumously. You have to be alive to get it. So, for example, Rosalind Franklin did not get. I can't even talk about Rosalind Franklin right now. Sorry. Yeah, also didn't get it. But all right. All right. In 1913, this individual, Edgar Adrian, British guy, this is a figure from his publication in which he first describes the all or none principle of an action potential. And he describes it as the action potential like a burning fuse. And here he's showing, in this figure, he's showing the action potentials in the central nervous cord of a caterpillar. At least he's not torturing cats. I was talking about brain electrodes in cats in the class previous to this one today. So caterpillars are a little bit easier to justify, I suppose. I don't know. Yeah, what you can see here, it's part of a chain of, so it's in the chain ganglia. A ganglia is just a collection of cells that in the human, a collection of cells outside the central nervous system. Do you need some tissues, Jay? That seems horrible. Blowing your nose on that stuff, that will make you not happy. Let's get you some tissues. Is that okay? I'm sorry. I've just been there before. They're all clean. Yeah. Yeah, it's the sniffly nose time of year. Yeah, so you can see the baseline electric potential in the ganglia. Oh, I was saying a ganglia. A ganglia is a collection of cells. Humans have brains, right? But then outside of the central nervous system, when you have collections of neuronal cell bodies, like we have the sympathetic chain ganglia that are outside the central nervous system, we call them ganglia. Okay, so but in a caterpillar, this is what kind of serves as a brain. They have this cluster of cells, and it's discharging. This is the baseline potential, and then you can see these like depolarizations, and then you can see the the lighter color, negative spike as it's heading towards the. So this would be like sodium potential. This would be minus 70 here. This would be maybe minus 90. This is plus 65 in our neuron. I don't know what the neurons of a caterpillar are like, what these actual numbers are, but you can see that negative spike is when the cell is going towards the repolarization potential, the equilibrium potential of the potassium. You can see that down here, and then it comes back up to the resting potential. All right. Next, a couple Germans, Neher and Sackmann, Erwin Neher and Bert Sackmann, a dynamic duo from Germany, and got the Nobel Prize in 1991 for some work in 1976, which was essentially the patch clamp technique, where scientists were able to measure ionic currents passing through a single ion channel. A single ion channel. Pretty cool. Pretty cool. So they had to, building on Barber, on the tradition of Barber 70 years earlier, they had to build a very special piece of equipment for drawing out these super tiny micro pipettes that were able to do this kind of work. So here's an example of what they would have done. They were able to characterize voltage-gated ion channels, which are going to be crucial to understanding the function of an action potential. So here's a neuron, here's an ion channel. They were able to put the micropipette over a single channel. And in this trace right here, you have this is just noise. That's stochastic flow, because there's always going to be fluctuations as things move around. But this is just the noise of a closed channel. Boom, it opens up transiently, boom, closes. So they were able to catch the opening and closing, the fluctuations of individual ion channels. So you see all these fluctuations, that are due to like incoming graded potentials that are summing and whatever, and then it hits this critical. It passes the threshold, opens the voltage-gated channel. It's open, repolarizes, and then is stationary again. Patch clamp recording was what they did. Okay, so we have all of the sort of historical pieces in place. Let's see how an action potential actually works. What are the step-by-step mechanisms that get it to work? Did they talk about all this in Josh's class? He talked about. Yeah, okay. Well, anyways, so this is an overview of it, and I'm going to go through it in detail. So it's a cycle, right? I showed you in that last trace that's a cycle. We're going to just start here at the resting state. It's chilling out. And then we're going to depolarize to threshold. The threshold is going to be minus 60 millivolts. This is because of local currents. We're going to open the sodium gate. Sodium's going to flood in. We're going to close the deactivation gate and open the potassium activation gate. And potassium is going to repolarize, and we're going to be ready once again. So I'm going to walk through those one step at a time. That was just a quick overview of all of it. All right, step one. Somewhere in the dendritic tree. Let's turn. Oh, yeah, I'm recording, which is exciting. Somewhere in the dendritic tree, there are synapses that are coming in with graded potentials, right? and there's local current. This can be from one graded potential, or it can be from multiple graded potentials that are adding up together, creating this local current and depolarizing this membrane. That like graded potential is spreading out, and this voltage gated gate is in what's called the excitable membrane. It's in the excitable membrane. So let's I should have a picture of this. Okay, here's a neuron, right? And the graded potentials maybe are coming in here. Graded potentials are caused by what kind of gate? What kind of gating leads to a graded potential? Chemically gated, chemical gated. And we get these local currents that add up. At this part of the membrane here, at the beginning of the axon hillock, the characteristics of the membrane change. They change, right? Now, this part of the membrane, we have voltage-gated channels. It's different. It's not chemically gated. From here, the axon hillock downward, it's electrically or voltage-gated, all right? So this local current's coming in, and there are sodium channels, right? Well, when we hit minus 60 millivolts, that causes the activation gate to swing open, and sodium runs down its electrochemical gradient. It wants to come inside because there's this huge chemical gradient of all the sodium that's outside the cell. Why is the sodium outside the cell? That's right. I'm going to get some mileage out of that one. Starts blasting in, going towards its equilibrium potential, which is plus 66, right? So in this diagram, we're just at plus 10 right now, but we're heading down. Just a kind of a fun little thing. Here's a sort of ribbon diagram or a space filling diagram of what these channels look like. This is a voltage-gated sodium channel, and there are these side chains that are that block the channel, right? And when the voltage changes, it pushes those side chains out of the way so sodium can fit through the channel. Turns out that this individual, Yoshizuma Tahara, was a Japanese person at the Tokyo Institute of Hygienic Sciences. In 1909, he isolated tetrodotoxin from the humble pufferfish. So the pufferfish is a delicacy, a common as a common foodstuff in Japan, but it has to be prepared correctly because the tissue, this like friendly little guy smiling at you there, you get close to it, it blows up, right? It gets big and puffy, and those spikes, you can see them. There's like coated in spikes. This one just wants to be friends right here. But if you make it seem like you're going to eat it, it spikes up, and it has this compound in those spikes. You get that, and it basically it interacts with these TTX binding, the tetrodotoxin binding sites on this little arm that is supposed to cover the sodium channel when it's closed, and basically keeps them open. So your sodium channel is open all the time, and all your nerves or muscles or anything that's been exposed to this, you just go like that. You just go into like rigor mortis. So, or tetanus is what it's called. It's called tetanus. It was like the It's not rigor mortis. It's tetanus. It's the freezing of the muscles. Just a little bit of a sidelight. Anyways, we're depolarizing here. And then at a certain point, when we get to minus 30 millivolts, so we didn't quite make it to plus 66. When we get to minus 30, the inactivation gate closes, right? So this gate on the sodium channel slams shut, like we're done. And at the same time, the activation gate on the potassium channel opens up. And what was the equilibrium potential for potassium? Say it again? Minus 90, that's right. And so we're at plus 30. So the chemical gradient definitely wants us going this way, right? Am I right or am I wrong? What direction is the chemical gradient pointing for potassium? Okay, why? The sodium-potassium ATPase is putting 2 potassium inside for every 3 that's putting outside. So there's a bunch of potassium in the cell, right? Hanging out with all the negative proteins. You can see them here. But the electrical gradient also wants to get it out of the cell. So potassium starts blowing out of the cell. A little, I don't know, historical tidbit here. This guy, Roderick McKinnon, in 1998, he got the atomic level structure of the potassium ion channel. This was actually pretty huge. It's not a trivial thing. They had a lot of, in 98, they had several soluble proteins, cytosolic proteins, that they had crystallized and got the atomic level atomic resolution structure of. But this is a membrane-bound protein, so for him to have gotten the atomic level depiction of its structure, which was crucial to understanding the function, the molecular level function of it, was quite an endeavor. His face is there. Undoubtedly, there was a giant team of people working with him to do that, but I guess he's the name we remember. Anyways, and then we head towards minus 90. At minus 90, the potassium gate closes once again, and we start the process over. All right. So when you think about an action potential, they are not only initiated in an all or none fashion. So like if you start it, it's started, right? But they are going to, it's like all of the excitable membrane. So if we start it right here, right, because it's coming here, all of this membrane is going to get excited, right? Every place that's got a voltage-gated channel on it is going to get depolarized and then go through this depolarization signal, all right? So that is an important point to make. Here's an example of what it looks like at subthreshold. You can get graded potentials that come that are going to depolarize, but if they don't make it to the threshold, then you don't depolarize. You don't get the action potential. So this would be like a weak stimulus. You get some kind of a weak stimulus, not a particularly strong graded potential, and you can create these graded potentials with an electrical stimulating electrode that is going to cause a local current. So then you can kind of increase that graded potential voltage that's impinging upon the axon hillock, and you're going to increase the number of times you make it to threshold, right? So now let's up the voltage, and now you see we get what's happening to the action potentials as we increase the stimulating current, the electric field. Basically, we're talking about creating an electric field in the body of the neuron. As we increase, ever increasing electric fields here, what's happening to our action potentials with time? They're becoming more frequent. All right? So we're just hitting threshold, depolarizing, repolarizing, but we reach threshold more rapidly because there's a stronger electric field. All right? We're able to depolarize again and again. So how fast can we jam these things together, right? How fast can we jam these things together? You can't actually have it be in continuous depolarization. There has to be this thing called the refractory period. It's a time period, it's the minimal time period during which a membrane that has gone through action potential is not ready for a new action potential. All right? Yeah. So action potential, and this refractory period is a consequence of the fact that depolarization in an action potential proceeds like a wave over the surface of the excitable membrane, all the excitable membrane, but it's a wave. It doesn't happen all at the same time. It's a wave. And the membrane that just went through depolarization, that's causing depolarization in the membrane downstream, that membrane that just depolarized has to go through the process of re-establishing itself. So if we look at how an action potential propagates over the membrane, say we have some depolarization to threshold at the axon hillock up in the initial segment. It depolarizes. We get it's graded potential with a local current that causes depolarization in this area to threshold, which causes a local current, and it moves like a wave down the chain. Here's a different way to look at it. So this is transmembrane potential along the y-axis and time along the x-axis. We're in one spot. We're just looking at what's happening in one place on the membrane. So we hit a graded potential, causes us to hit threshold. We're depolarizing in one. We hit threshold, sodium channels open. When the sodium channels are open and sodium's rushing in to the cell, you cannot initiate another action potential because you're in the middle of one, right? It's already depolarizing. So no matter, right here as this is happening, it doesn't matter if suddenly during that moment there is say you're sitting there and your favorite song comes on and you just have to bop. You're bopping, right? You're bopping, and the muscle that you're using to snap has started going. Meanwhile, in the middle of your favorite song, as you're just having a good time, a zombie werewolf senator from South Carolina or something like that comes in the room and is threatening to bite your head off or something. Who knows? limit your access to healthcare. And you're like, whoa! If it happens in the middle of this depolarization, it doesn't matter how much signal's coming into that neuron, right? It doesn't matter how much more electric field is coming in. It's not going to change what it's going through, right? It's not going to change what it's going through. If this neuron, this place on the neuron, this little piece of territory on the excitable tissue is going to have to go up here, let the sodium channels close, potassium channels open, and begin to reestablish and come back down into the range. All right. So there's this period we call the absolute refractory period. It's like no matter how much you want that neuron to like fire again, it's not going to do it. It's just not going to do it. Then there's this relative refractory period, right? Like maybe I'm playing some 20th century. I'm playing some free jazz, and you're having a hard time connecting with it, right? You're like, oh, this is kind of noise. I don't know. And then, but you're still like, okay, Dr. K, and you started a thing, and then I put on some Taylor Swift, and you're like, oh, thank God, finally some real music. And it happens, and you're ready to really start bopping. You can do it maybe during this relative refractory period. You could maybe go up again in here if you get a strong enough signal, if enough graded potentials are coming into that neuron, that you've created a strong enough electric field. During the relative refractory period, you can get it to go again, but it's not easy. All right, it's not easy until you get out of that relative refractory period. Then you're all good to go, whatever, and keep pericomo coming. Everybody understand? Anybody not confused? Like who's pericomo? Okay, all right. Albert Eiler, maybe better cultural reference. All right, so here is a different depiction, but this is a spatial depiction. All right, so here is the depolarization. This is in absolute refractory period. It's like in the middle of depolarizing. This just went through that, right? This just went through the absolute refractory period. And it's in like relative refractory period. This is going to cause this membrane here to depolarize. It can't go backwards because of this refractory period. So refractory period is necessary so that it happens across the entire membrane everywhere, but everywhere only once, right? Unless you have another signal, you reach threshold again. So it's not like constantly depolarizing all over the place, nonstop. This relative refractory period, that refractory membrane, prevents it from traveling backwards and allows it to travel across the whole membrane. All right? This is how, it's kind of like, imagine taking a rope. I got a rope stretched between us, and I go like this, and the wave travels between, from one end to the other. It's how the signal is able to translate itself longitudinally along the axon. It's just this wave of charge depolarization that moves across the membrane. This makes sense, right? I'm sure this, seems to make sense. Does it make sense? Does it not make sense? It should. I mean, it's kind of makes sense. But this notion of the refractory period flummoxed people for a very long time. They had a very hard time with it. So, hold on to your hats. Exciting topics. The history of the refractory period coming at you. All right, the first sort of the origins of the refractory period start early on in this hydraulic theory of what nerves were doing and how they worked. they had established, they had known Galen knew, because he certainly saw all these gladiators who were getting like hacked up. You could have a muscle, perfectly good looking muscle with a nerve going into it. You cut the nerve and then suddenly the muscle doesn't work, right? So how the hell do nerves work? He doesn't really understand electricity in the way that people would come to. So he thought that there was some kind of hydraulic mechanism in which the the nerves were somehow like forcing fluid, maybe from the blood, maybe some kind of humor, water, liquid humor was getting forced along the nerves and was kind of inflating the muscle. It's this hydraulic theory of how nerves work. And that was pretty much like problem solved for, like a millennia and a half. And then Rene Descartes said, well, maybe it's animal spirits that's coming from the brain. Because remember, he had this sort of like vitalistic, dualistic concept of the being as a machine, right? Like there's this animal spirit in you, but we're like everything, we're all little machines. And so maybe this like the animal spirit that's somehow living in the mental realm is flowing from the brain, because for him, you'll also maybe remember that it was the pineal gland was like the center of the soul. It was like the connection of the mind and the body, the mental and the physical for him. And he thought that it all descended through the nerves to these muscles to produce movement. So he's trying to like take Galen's idea, take it off the shelf, dust it off, and tries to reconcile it with his notions of like, animals and humans as little living machines. Next is this guy, Boerhaave. So, and we haven't talked about him yet. He was extremely influential. I think I had mentioned Boerhaave as one of the vitalists. But here's a quote from him in 1744. "The ultimate extremities of the smallest nerves," meaning like the motor end plates, the end of the nerves. Whew. Hello. My Lord. Earthquakes. Did it? Oh, geez, that is a sneeze. Come on, come back. Talk to me. Magic. Oh, no, come on. Normally that works. There we go. Is that right? No. There it is. Yeah, it's a heck of a sneeze. The extremities of the smallest nerves, deprived of their coats, hollow internally, being filled with spirits or nervous juice, derived into the nerve from its origin or fountain, the brain and cerebellum, by the continual force of the heart. Yeah, so, he's like mixing metaphors here for sure, you can tell. But there's a couple interesting things we can take from this. First of all, he actually, when he says the nerves deprived of their coats are hollow internally, he's talking about epineurium and like the connective tissue sheaths on the nerves, and he sees them kind of like tubes, and you put a cross section and you look at them. This is building upon the microscopic work that Van Leeuwenhoek that we talked about a little briefly. Maybe was that in lab or lecture? I don't remember when I talked about Van Leeuwenhoek. When I was talking about bacon and all that stuff. Anyways, or hook, not bacon. I'm just hungry, I guess. Yeah, he has a sense of the structure of the nerves, and he has a what's called a solidist theory of fibers that eventually would displace the notion of humors. This is in 1744, 50 years before Marianne and Antoine Lavoisier had finally, gave us a new concept of what elements were. But he also had the general flow of the brain and the cerebellum, etc., as being the source of this nervous impulse that was causing motion in the extremities. However, he believed that force originated in the heart, that it was the pumping of the heart that is somehow transmitted by the brain, because he knew there was a rich blood supply to the brain. He thought that somehow it was the heart that was driving this flow of fluids that was causing muscles to contract through the nerves as the pipes. And then there were these two, who were the students, the two most famous students of Borges, was Albert von Haller, was a Swiss guy, and then Robert Witt, who was Dutch. And they had quite an argument with one another over the actual function, sort of the inheritors of Borges. And the debate was over, in one telling, the irritability or sensibility of specific organs, right? That's what they would talk about. Is an organ irritable? Can you cause an action out of it? Or is it sensible and detecting things from the environment? So it was basically an outward or an inward facing organ. And the debate over the irritability or sensibility of specific organs was informed by the presence or absence of nerves in specific places. Like, is the liver richly innervated? Well, no, it's not, right? And with all of this, there was a debate about the spatial distribution, how this determined where the soul was in an animal body. And you can see with that the echoes of Descartes, right? This is all the echoes of Descartes' notion of the animal spirit as being that animating force that is being pumped through the nerves into the muscles to give them motion. So on one side, Albrecht von Haller he had a materialist view and of the function of nerves, meaning that nerves and muscles had some sort of inherent mechanical process, and that this was contrary to this sort of vitalist perspective. We've talked about this debate for a long time, right? The vitalist versus the mechanistic view of life, and these two were right in the thick of it. So here's a quote of his. "It is so certain that motion is produced by the body alone that we cannot even suspect any motion to arise from a spiritual cause." He's firing some shots across the bow at the church as well here. "Besides that which we see is occasioned by the will, and even in that motion which is occasioned by the will, a stimulus will occasion the greatest exertions when the mind is very unwilling." Meaning that he was paying attention. He was paying attention to some of the very earliest experiments on. This was prior to Galvani, but this was. I didn't go into the early history of the work that Galvani was based upon, but there was early evidence that if someone got shocked, that they were not able to control their muscle. You could stimulate a person either through their reflexes or some other external stimulus to contract a muscle outside of their will. All right. So that there's some kind of mechanism. How do you come to terms? How do you explain the reflex, right? A patellar reflex, for example. I do not want to move my leg. I can't. It's like very hard for me to not move my leg when I get that patellar reflex going. And so he's saying that there's some kind of mechanism behind it that is contrary to the notion of some sort of spiritual force that's causing action. That it's just a mechanistic view of life. For him, the refractory period was simply because that a nerve, there's still this hydraulic theory, this like premise that hadn't really been dispelled yet. And it's not going to be until much later, until they have a better idea of what nerves actually are and look like, until they're able to come to terms with the refractory period. But he thought it was because there's some sort of nervous fluid, whatever the juice is in a nerve, it gets depleted, and then you can't re-stimulate it. On the other hand, was this guy Robert Witt, Dutch, happy-looking fella here, who was a devout vitalist, and he totally out of hand said no, thank you to all the mechanical models that were being put forward, and thought that there was some sort of he was very Cartesian in this way. He thought that there was some sort of incorporeal agency, whatever that could be. That's God. It's like some, demonic whatever force. There's something like Descartes' demon. There's some thing that is controlling the body. And here's his words on the idea. We cannot but acknowledge that the all-wise capital letter author of nature has animated all the muscles and fibers of animals with an active, sentient, all caps principle united to their bodies, and that to the agency of this all caps principle are owing the contractions of stimulated muscles. So just like a classic vitalist perspective on muscle contraction. And as it relates to the refractory period, so this was simply based upon the nerve's exhaustion or fatigue. It's like susceptibility or openness to this external author, all caps, this divine influence or this vital force. So most of this, and when we're talking about this mostly in terms of the heart. Right? Because you may be thinking to yourself, how are they even thinking about the refractory period? Right? They barely understand how muscles work at all. Well, this was all related to the nervous innervation and the functioning of the heart. And it was recognized that the heart, there's like an upper limit to how fast your heart can beat, right? You can't just go like ba-boom, 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 like that. There is an upper limit to that. And so they wanted to understand what cardiac systole was. For von Haller, he thought it was it can't beat because it's sitting there waiting for more blood to get in there. Makes some sense to me, mechanistic view of the heart. Pardon me. Cardiac systole, the contraction of the heart, was caused by the force of the incoming blood. And diastole, that when the heart is at rest, wasn't able to happen until you got the heart empty of blood. So he had a very mechanistic understanding of the heart as a pump, right? Whereas Robert Witt believed that the heart could pump even if all of the he made the observation, I should say, that the heart could continue to pump even if all of the vessels surrounding it were closed, So if you take the aorta and the inferior vena cava and the superior vena cava, and you just close them off, the heart's still going to beat. So it can't be beating because of the force of the incoming blood or the release of the blood. Not like particularly fun if you're a heart, but that was a legitimate observation. Don't ask about how he was able to do that experiment on a living, beating heart. But, so that was a pretty conclusive refutation of the mechanistic view and a win, one of the later wins for the vitalist perspective before it finally went down the tubes. So, yeah, Witt wins that one. He's got a nice smile, but I liked Von Haller's wig more. Anyways, moving forward, you remember this guy, Felice Fontana? He observed that a small needle prick was all that was necessary to get a cardiac contraction. And so this was important because now he has an experimental technique. He has a method for initiating contraction, right? I can start a contraction by sticking a needle in it, some heart. I don't know. I forget what animal he was doing it on. And he made the further observation that if I do it again, it'll contract again. If I do it again, it'll contract again. I'll do it again, it'll contract again. However, if I do it really rapidly, there's a limit at which how fast I can do it. The heart will not beat. It won't go into contraction. There's this refractory period in which it is required for it to sort of reset itself. Well, it took over 100 years before this was really described as a refractory period by Francis Gotch, a British dude, who coined the term refractory period right at the turn of the century. So that we're jumping ahead 100 years because there wasn't a lot of progress made on the concept of refractory period. And I don't know why I'm putting everything in these like dichotomies of people today for you, but I am. These are two people that we've seen already. We've seen Albert von Kolliker, a Swiss guy, and Robert Rimac, a Polish individual. And you may remember Rimac, for example, had some of his ideas scooped by Virchow. And it was a Polish Jew that really had to deal with a lot of problems. So we're going to talk about some of that right now. Anyways, in 1896, Kolliker coined the term axon, and Rimac was able to describe myelinated and unmyelinated axons. Okay, so there are like two little data points that we have that help us on our way towards elucidating signal transduction. But I want to go into some. I want to go into some sort of facts about the two of them. Two geniuses in their day. Two very well-respected. Well, I shouldn't say that. Two acknowledged geniuses. Kolliker, I think I've said this before, pretty much every tissue in the body, whether it's the liver, the heart, the brain, muscles, nerve fibers, on, he studied all of them, and some of our foundational knowledge about all of them comes from this guy. In 1889, the histological preparations that have been done by Ramon y Cajal. I think I had talked about him a little bit so far. I haven't gone into Ramon y Cajal in depth yet, though, have I? No, I haven't talked about him at all. That was the other class. I talk about him a lot because he's important. Well, we're going to learn about Ramon y Cajal in a little while. But he made these exquisite, beautiful drawings from these really incredible histological preparations and answered the question of a different debate between reticular formation or neuronal formation in the brain. We'll get to Ramon y Cajal and that later. But he went on to do that, which was important because it showed the reproducibility of Ramon y Cajal's work. He also was the author of saltatory heterogenesis theory. And saltatory heterogenesis theory merits a little bit of time because it was an important concept that has its roots in racist ideology and was definitely an important sort of cornerstone for scientific racism. And it's the notion that a couple things that sound pretty science-ish, right, that are related to things that Darwin said, which is that things evolve, right? Things evolve, natural selection. And saltatory means start and stop, like start and stop, like fits and starts. Something that's saltatory is like fast, slow, fast, slow, fast, slow, like that. And hetero means different, genesis, origins. So in his theory of saltatory heterogenesis, that there were multiple origins of living forms. That certainly jellyfish are in no way related to human beings, which are in no way related to plants, which are no way related to like the tiny little critters that are prokaryotes that we see swimming around under a microscope. They all evolved, they're different origins. It was like impossible for this guy to conceive of the time scales necessary for life to evolve from a single origin, right? Like these biologists at this time had a hard time wrapping their heads around how ancient the Earth is and how much, like, what a billion is. Even today, when I think of Elon Musk, it's hard for me to think of what a billion is. It's kind of obscene, actually, but you can see right away where this guy is about to drive off the map. I mean, it's fine to make like a mistaken presumption like that because you have a hard time coming to terms with how ancient the Earth is. However, you can also see how this could easily be used to claim that humans had, like the different races of humans, all had separate origins. So people of African descent, Asian descent, Caucasian, what was later termed Caucasian European descent, all had separate origins. And that was part of this saltatory heterogenesis. And he said the evolution happens, it just happens very rapidly, and then it's kind of like where we're at. So humanity is at this like stable state right now, and it, can jump forward in rapid periods of time. And the Europeans were going through one of those rapid jumps in evolution that was putting them beyond other species of humans in the 19th century, according to this telling. Anyways, people loved this guy, at least the rich people that were running things, like Prince Regent Leopold of Bavaria, and gave Albert Kolliker the prefix von. If you know people that have von or van in their last name, that it's like Sir in, like, Sir so-and-so, Sir Winston Churchill in British. They've been knighted or whatever. It's like a term of nobility, of noble recognition. In their family, it gets, it doesn't have to be the person that has that. It gets passed down through the family or whatever. So he became von Kolliker. And he was awarded the distinction and honor of becoming a fellow to the Royal Society in 1860. And 1897, he got the prestigious Copley Medal. And there's even a species of lizard still named after him, Hyalosaurus caulacari, named after this guy. So he comes up with this, like, he studies a lot of stuff in tissue, comes up with a racist explanation for life, and then gets a lot of awards. Short summary for him. Let's compare him with a no less brilliant individual who happened to be Jewish. So we've already covered how Rudolf Virchow stole his idea that all cells come from pre-existing cells, right? It's like cell division theory. Rimac came up with the basic idea of cell theory. He also experimentally confirmed Louis Pasteur's unverified assertion that there is no such thing as spontaneous generation of life. Life doesn't just like come out of nothing if you wish real hard. I mean, it happened once on Earth, but it hasn't happened multiple times. This is so Pasteur and Rimac, they were on the other side of this saltatory heterogenesis argument, right? Saltatory heterogenesis would have you believe that life can start multiple times because you have to have that. That's like a necessary ingredient to be able to say that this person with this skin color or these facial characteristics is not in any way conceivable related to me, right? So, Rimac said, well, yeah, there's a lot of magic thinking in that hypothesis. It requires spontaneous generation of life. Pasteur hypothesized that was incorrect, and Rimac actually experimentally showed that was not the truth. He also identified 3 embryonic germ layers, ectoderm, mesoderm, and endoderm. In so doing, basically established the field of developmental biology. And I mean, we're talking about major innovations. His name should be as familiar to biology students, as Louis Pasteur, at least, if not as much as Darwin. But despite all that, he was never to become a full professor. He was denied that because he was a Jew. And he was the first Jew that was able to teach at the University of Berlin after a long life and only being able to become an assistant professor, never an associate, never granted tenure. And his son and his grandson both went on to be scientists and mathematicians. His grandson was actually a very accomplished and now well-regarded mathematician, Robert Rimac, who died at the hands of the Nazis in Auschwitz in 1942. So just a kind of a case study in how prevalent anti-Semitism was at the time in the 19th century in Europe. And I'm sorry.